Okay, thank you very much. Thanks, Sebastian. And I would like first to thank the committee for selecting my work and inviting me here. So again, my name is Alessio Buccino, and I'm a PhD student at the Center for Integrative Neuroplasticity at the University of Oslo, under the supervision of Gaute Einevold, Philip Affliger, and Marianne Finn. And I also collaborate with Gert Kauenbergs from the University of California, San Diego. And today, my talk is about a computationally assisted approach to neural electrophysiology. So it's a different kind of electrophysiology. I'm going to talk about in vivo extracellular electrophysiology. And my talk is mainly divided in three parts. I will first describe the biophysical modeling framework that we use for basically this computationally assisted approach. And then I will spend most of my time explaining some application in the electrophysiological pipeline that we can improve with some computational-based and data-driven approaches. And finally, I will show some limitation of the current modeling framework and how we can address it with more advanced modeling, and also some improvements and future directions. So let me start with this beautiful slide from artist Gregory Dunn, which shows a slice of brain tissue, and we can appreciate how different types of neurons are present, and they build up neural circuits by connecting with each other and by basically communicating their activity in this spatially organized tissue. And in systems neuroscience, the way that we can probe this activity quite simply is by basically inserting penetrating electrodes in the neural tissue. And for decades, researchers have been using these tetrodes or microwires, which are basically just metal wires that we can insert in the brain, and they can pick up the activity of a few neurons at a time. But in the latest years, there's been a huge development in another kind of probe called silicon probes or multi-electro arrays. For example, these neuropixels probe, now it's going to be very widely used. And these probes have hundreds of recording sites, and they can pick up the activity of many different neurons at a time. And also, they provide a much higher spatial resolution. And we also know the geometry of the probe, so we can actually adapt our electrophysiological techniques to include this spatial information. Rather than with these tetrodes or microwires, we don't really know the position of the electrodes, so we cannot really use spatial information. So I'm going to use mainly, or I've been using mainly, a multi-compartment model developed, I guess, next door from the Blue Brain project. And these models are mainly consist of two different parts. The first part is the morphology, and you can see it's very detailed, and it's extracted from beautiful microscopy images. And the other part is the electrical properties, which are extracted by fitting some electrical parameters of the model to patch clamp recordings. And when we have these very detailed models, we can simulate the extracellular activity by combining the solution of the cable equation, using, for example, the neuron software, and then we can use volume conduction theory to compute the extracellular potential in any point in space. And in that way, we can simulate the activity of these neurons on these multi-electro arrays that I showed you before. These models to me look very realistic, and I couldn't see any difference between the simulations and the recordings, but don't trust me. Some main experts in the field basically had this very nice opinion paper in which they say that these models are so complicated and so detailed that it would definitely pass a biological Turing test, as long as the experimenter wouldn't go and check for single channels dynamics, because these are conductance-based, so they have basically an average distribution of channels. So how can we use those models in order to improve the way we currently do electrophysiology? Let me first start. Oh, now it started blinking. Okay, anyway, it's okay. It didn't like my black slide.
We had this morning already, yeah?
But now it's going to work now. I'm confident.
No.
Is that okay? A bit flashy, but it's the end of the day, so it's good. Might keep you awake. So this, what I mean with the electrophysiological pipeline. So we start with these raw signals, and then we usually filter them to extract the high-pass filter activity. And then the first part is spike sorting. And I will talk a bit more about this, but it basically attempts at extracting the activity of single neurons firing from the recordings. Once we have sorted the data, we can extract the average spike waveforms, and then we can use these very rich information to localize and classify neurons around the probe. And then one might want to also use close the loop. And then I will also show how you can design some optimized stimulation paradigms based on what we extracted in order to, for example, induce neuroplasticity in vivo by stimulating a recording neuron every time it spikes. So what can we do for spike sorting? So again, spike sorting is this process of extracting the activity of single neurons from this mixture of extracellular recordings. And it's unsupervised in its nature, because when we use these penetrating electrodes, we cannot really see what we're measuring. So we need a way to extract this information. And it's not a trivial problem. In fact, so many researchers have tried to solve or to attempt to solve this problem. And only that I know of, in the last 2 years, there are at least 1, 2, 3, 4, 5 spike sorters, so many that the researchers have started calling their spike sorter yet another spike sorter. So my idea here and my contribution is to use computational modeling to try to create simulated, biophysically realistic simulated recordings that can act as a referee for this spike sorter to show which one is better in different conditions. So we use these biophysical simulations from these realistic cells, and we built many of them with different positions and rotations among the multilateral array to build these spike templates. And then on the side, we can simulate different spike trains, for example, with the Poisson or gamma processes, or we can also use spike trains extracted from real recordings or from biophysical network simulations. And then the idea is that we can very quickly and very easily build our basic trace by convoluting these spike templates with the spike trains. And here we can also apply some amplitude modulation. Which could also include some physiology information. For example, we could modulate the amplitude based on the bursting index of the spike trains. And then we can also add a noise model which can resemble the noise that we measure with experiments. So the noise can be, for example, correlated with the distance between the electrodes or fully extracted from experimental recordings. And this... Okay, anyway, so basically the contribution here was building a open source simulator that spike sorter developers can use in order to test their algorithms in different conditions. So here everything is controlled, so one could easily control the number of cells, their position, their overlapping. It could control the synchrony of the spike trains, so some units could also be spatio-temporally overlapping, which is very hard for spike sorter algorithms, and also the level of noise can be easily controlled. And now I should have had a video, but we didn't find a way to make it work, so imagine. Okay. So you can see this spiking activity distributed over different electrodes, and I think these kind of recordings that are highly customizable and very fast to compute could be a really good addition to the field in order to test the different spike sorters. So after we have spike sorted our data, we can easily compute the spike waveforms with the spike trigger average. And then I spent quite a bunch of time during my PhD trying to figure out a way to improve localization and classification of cells from their extracellular signature. So the usual way to approach localization is by trying to solve the inverse problem. So imagine we have one, here it's 2, but imagine it's 1 extracellular action potential. And then in order to solve the inverse approach, we need to choose a very simple model. For example, we can assume that a neuron peak is basically made from a single monopolar current source in space. And then we can try to minimize the error between what this simple model would give and what we observe by moving this single current around until we actually minimize the error and we find the estimated position. But the main problem of this approach is that we need to use very simple models to describe the neuron, because otherwise we would have too many parameters to estimate. And these neurons are not so simple. They're very detailed and the dynamics is very rich. So instead of this unsupervised approach, we decided to use a supervised approach. But the problem here is that it's very hard to obtain ground truth experiments. So it's very hard to measure the location of a neuron that you're recording in vivo. So since we have so good realistic models, we use basically these simulated data in order to build large data sets of extracellular action potential that we use to train our model. From this extracellular action potential, then we do the usual machine learning pipeline. We extract some features, but since we have these two-dimensional multi-electro arrays, we extract feature from each of the spike and we build these feature images. And since we have this 2D distribution of features, then we use convolutional neural networks in order to extract from these feature images. And for localization, we use the peak, the negative and the positive peak images in order to basically predict the X, Y, and Z position of the soma. And how does it compare to state-of-the-art algorithm? So here I'm showing the error between the ground truth position of the soma and the reconstructed one. And we can see that the convolutional neural network performed way better than the other inverse approaches. And you can see it also here in the cumulative distribution function. And we were able to reach an accuracy below 7 micrometers, which is highly acceptable for extracellular recordings, considering that the soma of a neuron is 10 to 15 micrometers. So we were very happy about that. And we also use the same approach to solve neural classification. And neural classification usually is approached in a different way. So the spike with the largest amplitude is selected, and then two features from the spike waveform are extracted: the peak-to-peak width and the full width al maximum. And pulling all the spikes from different experiments, you can plot each spike in this two-dimensional space with width and full width al maximum. And then you would use clustering, so an unsupervised technique to find two clusters. One for excitatory cells, which usually have a broader waveform, and one for inhibitory cells, which have a narrow waveform. The problem here is that the distinction here is not so clear, and I'll show you later. But we used exactly the same approach also to solve this problem, but here is a classification problem. And we used the same feature images, but instead of the peaks, we used the full width al maximum and the width of the spike. And instead of predicting X, Y, and Z, we predict whether a cell is excitatory or inhibitory. And then we compared with this unsupervised approach. So here I'm showing each of these points is a spike in this full width of maximum width space. And these are more than 10,000 observations. And it's true that excitatory cells in blue on average have this larger waveform, but there is a large region of overlap. And standard clustering techniques cannot untangle this overlap. And we tried k-means and the mixture of Gaussians. Instead, when we use convolutional neural networks, which use all the rich spatial information of the extracellular spike, we're able to untangle the overlap, and we reach a much higher accuracy, around 98.4%. So finally, I'm going to show another application in which we use a model-based approach to optimize stimulation patterns. In this case, we need a model of stimulation from extracellular potential, and we use the activation function, which predicts the excitability of a neuron based on the second derivative of the extracellular potential. And then we can compute the potential given by the currents from the different electrodes using simple volume conduction theory and the semi-infinite plane approximation. And instead of using the standard monopolar or bipolar or multipolar approaches, we set up an optimization problem. So assuming that we know the soma position and the axon hill direction of the neuron, we use the genetic algorithm approach in order to optimize two different fitness functions. So the first function aims at improving the selectivity of the solution. So it's basically, it's high if the target neuron is activated and the surrounding neurons are at rest. And the second fitness function basically gives us an energy efficient and sparse solution. And this is an example of one of these scenarios. You can see the target neuron in red, and the gray neurons are the surrounding neurons. And we can see how these different currents in the multi-electron array shape the extracellular potential so that only the target neuron here in red has an activation function above a certain threshold, while all the others are safely below. So we tried 1,000 different scenarios with random positioning and direction of the neurons, and very quickly the genetic algorithm approach has a much higher specificity, and it only elicits spurious spikes on non-surrounding neurons in a very few cases compared to monopolar and bipolar approaches. So I hope this doesn't screw it up again. So far I use a very well-established modeling framework, but it has some limitations. So in the last few minutes, I'm going to show this limitation, and I'm going to describe some new results that we've been working on in order to improve them. So I'm not going to go into the limitation of the cable equation because this is only important when you consider the extracellular interaction between two neurons which are close by, which is so-called ephaptic effect. But I addressed this assumption. So in volume conduction theory, you assume that the extracellular space is infinite and homogeneous, and this is not clearly the case because we are using a neural probe to pick up the signal. So we used a finite element approach in order to explicitly model the presence of the probe in the extracellular space. And we basically modeled the dynamics of the neuron, the intracellular and extracellular space separately. And basically, we investigated the effect of three different kinds of probe: a microwire, and these two silicon probes, a NeuroNexus commercially available probe and these NeuroPixels probes. And what we found is that there's no difference for the microwire, for which the recording is at the tip. There is a large difference for these multi-electron arrays. And the amplitude of the spike can be almost twice as large as the spike without the probe. So we're now working in trying to correcting these amplitude gains with a probe-specific and electrode-specific approach. So finally, I showed you all these very nice, in my opinion at least, computationally assisted approaches, but they're not yet validated, and this is because validation is extremely difficult. So there have been some attempts that try to basically measure the same cell extracellularly and juxtacellularly with a glass micropipette, but it's hard to estimate the perfect location of the neuron. So now, in the rest of my PhD, I'm trying to combine a planar multi-electron arrays with two-photon microscopy in order to basically being able to electrically imaging the extracellular potential of the electrode, and at the same time using imaging in order to see the position and the cell type of the probes. And this computationally assisted approach finally could be used to drive the design of new probes, in which we could use these in silico models for finding the best spacing or the interface, the geometry of the electrodes, or the configurations of the electrodes, and so on. So in summary, I showed you different applications in which we can use a model-based and data-driven approach in order to improve electrophysiological recordings, and hopefully this is going to improve the methods that we use in order to understand what our brain does. So I would like, oh, it's not here, I had an acknowledgement, probably it's in the other. I would like to thank my group here. It's there, I didn't forget. Yes, there you go. So I would like to thank all my collaborators, my group in Oslo and in San Diego, and the Simula Research Laboratory for funding my PhD, and also some colleagues in Freiburg for collaborating with me. And I'd like to thank you for your attention, and I'll be happy to take any questions.
Thank you, Alessio, and apologies again for this projection problem. You handled it very well.
It's okay.
Questions from the audience?
Very interesting talk. Thank you. Could this research be commercially interesting for some companies? Do you have what kind of companies could be interested?
I think we just wrote a grant in which we had to find some industry partners. And we reached out for a company that builds these high-density planar multi-electro arrays for slice and in vitro recordings. And I think these kind of methods could be used, for example, for automatic localization of neurons and placement of micropipettes for patch clamp recordings. This is one possible industry outcome. Yeah.
Thanks, Alessio. Very interesting. Two questions. One, it's about the validation.
Mm-hmm.
Because the validation per se could take forever.
Yes.
So what is the idea when there is the criterion of the validation becoming okay? So when you want to stop the validation?
Yeah, so the main idea behind this is to acquire a data set in which we know from the electrodes that we are, for the neurons that we are recording, we can extract the exact 3D location with respect of the multi-electro array. And that would be able, with this kind of recordings, we would be able to validate the localization approach. And also we could use some sort of cell tagging, targeting only excitatory or inhibitory cells, so that we could target the classification. But yeah, I mean, this goes way beyond, so we could also improve the models with this kind of combined and simultaneous recordings. But I think we have quite a well-established modeling framework. Of course, the models will be a little bit different in case of the planar array, because we would need to use a semi-infinite modeling.
Another question on the modeling.
Yes.
There have been few works already on hybrid models at different levels,
Yeah.
cortical, spinal, peripheral, whatever. What is the novelty of your model with respect to the previous ones?
Yeah, so
This one.
this is a full finite element. I think it's a kind of an overkill for this application, so we could have used a hybrid approach. But basically we have some, this model has been developed at Simula, so with some collaborators we are just trying to find some application in order to use it. I think we would get the same results by substituting the neuron with just the transmembrane currents and use the hybrid approach in this case. But now we are actually using the same model to simulate electrical stimulation. So in that case, for example, this could be like, this model could be interesting because one could study, for example, the shadowing effect of the soma on the axon hillock. And that could not be studied with the conventional cable modeling.
If I have very last question, you showed in the genetic algorithm two different fitness criteria.
Yes.
They were combined, or they were one or the other? And if they were combined, how you combine them?
Yeah, they were combined, and now I don't remember exactly the coefficients, but I think it was basically they were equally weighted. So it was optimizing for both fitness functions at the same time.
Okay, we have to move on. Thank you, Alessio, once more.